All right, thank you very much for giving me the opportunity to make this presentation. My name is Hussaini, a researcher with the National Veterinary Research Institute, VOM, and it's a pleasure to make such presentation to this great audience. By introduction, foot and mouth disease is an endemic transboundary animal disease in West and Central Africa, and the disease affects all domestic animals, including wildlife, those that are cloven hoofs. And it is one of the major diseases that impact negatively on trade of livestock and livestock products within the region. Foot and mouth disease is actually belonging to aftovirus of the family Pequinaviridae. And we have 7 different serotypes that actually exist. And these are serotype A, O, and C. We have Asia 1, South African Territory 1, 2, and 3. And multiple subtypes occur within each of the serotypes. In West and Central Africa, we belong to the pool 5 of this particular disease. And to date, we have 4 circulating serotypes within the region. The regional pools, as I said earlier, we have about 7 regional pools, but specifically we are talking about West and Central Africa, which belong to pool 5, and the viruses circulating in this pool 5, as I said earlier, is type O, A, SAT1, and 2. Though there are claims that serologically, particularly in Cameroon, serotype 3 of South African Territory, SAT3, has been serologically identified in Cameroon. So the disease is characterized by fever, that's the clinical sign, and lameness. You have vesicular lesion on all the hairless area of the animals, particularly the tongue, the feet, snout, and in the teeth region. The disease is highly infectious and can spread easily through contact with infected animals and contaminated material, such as formites and some in cases where there are outbreaks that you see the personnel going from one farm to another without proper decontamination. They could serve as a source of transmission of this particular infectious disease to other farms. And vaccination and movement control are vital instruments for the control of this disease. This is just a picture showing one of our outing, field surveillance, and this is an active FMD outbreak case that we went out for sampling, and you could see this erosive nature of the tongue caused by foot and mouth disease. And this particular lesion was actually the tree of the infection that we were lucky to collect sample from such animal. So what about the epidemiology of this disease in West and Central Africa? In fact, it is something that is very challenging, that the epidemiology in West and Central Africa is very complex, So effort to control the spread of this virus in this region is very complicated. And the reason being that we have ineffective border control, unrestricted movement of the nomadic Fulani between neighboring countries, and of course, lack of vaccination campaigns among the African countries, particularly within West and Central African region. Then we have limited knowledge of even the current circulating serotypes and topotypes within the region. So what can we do? So there is also a need, therefore, to develop a vaccine that will incorporate the serotypes and the antigenic strength that is circulating currently in the region. Let me get a point across to you people that currently there is an incursion of East African topotype 3 that phylogenetically is traceable to the East African topotype from Sudan, East African topotype 3, that was actually detected in Nigeria. So, as I said earlier, the pool 5, you could actually talk about the epidemiology, that is just a hypothesis, how these viruses are transmitted across the region. And I want to say, particularly in Nigeria, because of the market in Nigeria, the population in Nigeria, so most of African countries that have cattle migrate to Nigeria for the sale of these animals, and some of them are actually coming for greener pasture like feed, water point, and Nigeria end up as the places where most of the African countries migrate their animals to, and a huge population of animals moving from places like Benin, Niger, they all end up in Nigeria, and you have some border countries like Nigeria and Mali to Burkina Faso. All this movement, as they move across country, they transmit these viruses. And of course, I earlier mentioned we don't have restricted borders. Our veterinary services are actually not too strong, and therefore we end up having challenges of these viruses crisscrossing across the nation in this region. So we talked about the risks. There are zones that actually are risk zones. And you could see from this map, the area that is highlighted in red colors, where you have high animal densities, and their movement are related to trade and pastures, and also water points, as I earlier highlighted. And I said Nigeria being the country that is most populous in the region, and of course the number of cattle in Nigeria is not compared with the number of cattle across other nations like Sudan and Niger. So they move their animals for trading into Nigeria. So because of this, there are main risk factors. And these risk factors are what? No existing quarantine at the borderline. We don't have that. And we don't have effective checkpoints in this borderline. In Togo in 2016, it was advocated that maybe the region should have like a panel of moving animals across the border, which is something that needs actually to be considered. And you see, uncontrolled movement of personnel during FMD outbreak, which I said could be as a source of fomite in transmitting this disease, across the farms. You see, and also we have the weak capacity of our veterinary services. I want to tell you that the problem we are having in this region is that we don't stand to our responsibility as veterinarians. And just to say that we don't really pursue this disease, and we don't really pursue to know about the epidemiology of this disease in this region. And also to mention that the disease is basically ravaging animals, and the Fulanis are helpless, but we are not really providing solution to them. And also lack of good communication within the countries of the region. We don't communicate. Since the meeting in 2016 in Togo, I don't really know if the sub-regional groups set up during the meeting are really communicating, but that was the mandate that was given by GFTAG, that we should communicate within the region to know actually the problem that is happening concerning FMD within the region. So, just to talk about samples of interest in terms of isolation, in terms of serology, we may be using ELISA. In Nigeria, we basically go after the vesicular fluid. If we cannot get the vesicular fluid within the 3 days of incubation period, we target the fresh epithelial tissue, and we will get serum sample just to identify whether these animals have antibodies to FMD. And fortunately, when you use 3ABC, you could differentiate whether this is infected or not infected, if it is a vaccine antibody. But I don't even know in the region which of the country is even vaccinating against the disease. So, and where we cannot get serum, and in a milking cow, we normally take the milk or nasal discharges for RT-PCR. And on my right, you see all the required tests for you to diagnose this disease. A virus isolation, using in Nigeria, we are using a cell culture line called ZZR, and we use 3ABC ELISA, and the VNT is a technology that up till now we are not using it because of you have to work with the live virus, and we don't have the facility to contain the live virus. So the RT-PCR and sequencing, we normally use the Fabric Institute for sequences of our isolate. Yeah, the regional control situation of FMD. FMD control is just like the progressive control pathway is an evidence-based tool by GFTAG and OIE to evaluate the progress made in the control of FMD globally. Unfortunately, most of the West and Central Africa, the PCP stages, none of the country has gone beyond PCP stage 1. So this tool is actually used in order to control FMD situation in the country. So the current status, according to the progressive control pathway, as you can see, you see the blue country, the map showing. You can see in blue countries that are actually free, and I recognize free without vaccination. The ones in light blue are countries that are actually recognized by OIE to be free with vaccination. And the one that is sky blue, official control program, yes, they had endorsed, the OIE had endorsed the official control program. Then the ones you have red, because of one challenge or the other, they suspended the status, the free status of such country, like presently what is happening in South Africa. They are placed under suspension because of the recent outbreak recorded in South Africa. Then countries and zones without FMD official status are the ones in gray. You could see countries that are actually showing gray colors are the ones that they don't have official status of whether they are free or not. So this is a PCP situation in Central Africa. Let me just go to the next slide. You will see, as I have actually put it, there are 4 countries in Western Central Africa that are on stage 0. These are Cape Verde, Ghana, Liberia, and Sierra Leone. So these countries were assessed as stage 0 due to the absence of control measure on non-circulating strength, and the epidemiological situation is not known in that country. Then we have 7 countries which we are professionally assessed as stage 1. These are Benin, Burkina Faso, Cote d'Ivoire, Guinea, Guinea-Bissau, Niger, and Togo. All these countries are given the provisional status. So these countries, they actually provided evidence of compliance to stage 1 requirement, which including the assessment plan and the identification of the circulating serotype. And what I'm presenting to you is actually what was presented in 2016 in Togo. So 4 countries within the region, that is Gambia, Nigeria, Mali, and Senegal, complied with the minimum requirement for inclusion into stage 1. So based on the self-assessment questionnaire and the information provided during the meeting. And let me, at this point, I just want to pose a question here. So, because in West and Central Africa is actually a challenge that we don't even know the role of wildlife in the epidemiology of this disease. And simply because we claim that we don't have buffaloes, and I am here presenting this question for the group to discuss whether somebody have information on the role of wildlife and the epidemiology of this disease in West and Central Africa. And thank you very much for listening. And I am here to listen to some of the arguments that will be generated from this presentation and the questions that will follow for me to respond to them. Thank you very much.